Thank you so much for the introduction, Ian, and to the organizers for inviting me to participate in this virtual symposium. This is amazing. So today, I'm going to talk to you about some ongoing work in my lab that studies host microbe interactions in the vaginal tract. So aside from there being host and microbes, this is a really complex interaction that we don't understand a whole lot about. And when we talk about the microbes of the vaginal tract, certainly there are commensal microbes, but there's also these pathobionts, which are organisms that are present in the absence of disease, but are capable of causing diseases should the host gain some type of susceptible state. And so two of the pathogens, as mentioned in the introduction, that I study are group B streptococcus and extraintestinal pathogenic E. coli. These are both very similar organisms in terms that they colonize the reproductive tract, including the vaginal tract, and can disseminate to cause invasive disease, either in the neonates or in the urinary tract. And so, of course, one of the things that we're interested in is studying virulence factors that these different organisms have that can cause disease. And then we're interested in understanding what the host immune responses to these organisms are. But it gets more complicated because, of course, there is this endogenous vaginal microbiota that can also mediate pathogen colonization or resistance to pathogens, which would be a beneficial effect. And then we can further complicate this by starting to look at host metabolism and how that might interplay into all these different interactions. And so, of course, host metabolism can impact the immune response. We're particularly interested in understanding how the host metabolism can alter the virulence potential of some of these pathobionts. And of course, we're also interested in understanding how altered host metabolism can mediate the composition of the vaginal microbiota. And some of the focuses of my lab in particular are host metabolic states that are unique, that impact the reproductive tract, including pregnancy, metabolic disease such as type 2 diabetes or gestational diabetes. And then we're also interested in understanding the role of the vaginal microbiota in autoimmune disease. And so I know we'll touch on this probably multiple times during this session. I just wanted to give a brief bit of background on the vaginal microbiota of humans. And so the vaginal microbiota is a unique microbiota compared to other body sites. It's typically dominated by a single lactobacillus species. Most often, this is either Lactobacillus crispatus or Lactobacillus inners. However, there's this other community state type, or CST, that instead of having a dominant lactobacillus species, tends to have a mixture of other facultative anaerobes. So this can include Gardnerella, Bifidobacterium, Prevotella, et cetera. And it's unknown why, but it's definitely widely clinically supported that individuals with this community state type 4, this more diverse community state type, are at an increased risk for adverse obstetric or gynecologic outcomes. But that really raises the question, are these causal in this increased risk, or is this merely an effect of an altered host state? And so this is really a question that we're asking in the field. And so to understand the functional role of the vaginal microbiome in human health, we really need high parameter longitudinal studies that really assess the entire vaginal microenvironment. And then we also need to pair this data with experimental model systems that reflect the human vaginal microbiota. And so for this talk today, I'm going to focus primarily on the second point, which is some of the work that we've been doing to make some better, more reflective of the vaginal environment model systems. And so the first that I'm going to introduce to you is an in vivo model. And so it's widely accepted that in the gut microbiome, germ-free or notobiotic mouse models have been instrumental in advancing what we know about the gut microbiota in health. Unfortunately, it's also well known that the murine vaginal microbiota is not reflective of the human vaginal microbiota. So a similarity is that they do tend to be dominated by a single vaginal taxa or bacterial species. But unlike humans, this tends to not be lactobacillus. So instead, mice tend to have staphylococcus dominant or enterococcus dominant communities. Lactobacillus is occasionally present, but this is quite rare, less than 10% of animals. And this data is from a standard lab model, wild type C57 Black 6 mice from Jackson Labs. So we've tried a variety of different attempts, and I'm happy to discuss this further in the discussion, to try to humanize the vaginal microbiota. And so I'll tell you one of the most successful routes that we've tried And this is to take a humanized microbiota mouse, which is germ-free mice that have been colonized with human-pooled fecal microbes. And this is a mouse model established by our collaborators, Dr. Britton here at Baylor. And what they've shown is that these mice tend to have a more human-like gastrointestinal community. So we also assessed if their vaginal microbiota tended to look more human. And so what we found is that the most common community state type in these humanized microbiota mice actually had a lactobacillus as the dominant organism, a Lactobacillus gasseri, in fact. However, an important caveat is this, of course, would be a human gut gasseri strain and not a vaginal strain. We also saw some community state types that looked more like the mouse, so a Staphylococcus or Enterococcus dominant community state types. And then we also saw Oligolactobacillus community state type. So it's not perfect, but it's definitely a step in the right direction. And we asked whether or not this mouse model showed any different phenotypes in terms of pathobiont colonization. So we challenged these humanized microbiota mice with either group B Streptococcus or Prevotella bivia and assessed reproductive tract colonization in non-pregnant mice. And consistently what we saw is that the humanized microbiota mice showed decreased bacterial colonization levels, suggesting that the more human-like vaginal microbiota was protective compared to conventional mice. And so we're interested in understanding more about how these microbes are mediating this effect and what role the immune response plays. But of course, these are not perfect models, and we really want to understand how vaginal microbes themselves are interacting with some of the pathobionts that we study. And so we've also begun to develop models that we can use to cultivate vaginal communities in vitro. And so we've adapted a continuous flow bioreactor system, again, developed by our collaborators, Dr. Rob Britton here at Baylor, that basically uses these strips of 6 bioreactors that have about 10 to 15 mils in volume. And you can use continuous flow, so there's constant media in and waste out, to establish stable communities. They're also fairly small scale, so we can put this entire apparatus into an anaerobic or microoxic chamber. And the data I'm going to show to you, this whole model system has been brought about by 2 talented grad students in my lab, Sammy and Claire. And so what we found is that in these bioreactor systems, we can cultivate a level of bacterial density that's similar to what we see in human vaginal fluid. And so this can be a culture density that we can achieve over multiple days. We use a simulated vaginal fluid with several adaptations. We use microoxic conditions to mirror the oxygen levels of the vaginal tract. And then we use basically a slow of a flow that we can to achieve a similar media changeover relative to the vaginal environment. And what we can also see in these communities is that they are able to drop the pH down to levels seen in the vaginal tract, again, suggesting microbial growth and glycogen fermentation. And this is data collected from 6 healthy human donors, vaginal communities collected off of swabs. And what we can see in terms of composition of these communities, when we assess the original input, so what the bacteria was on the swab sample, and look at our communities that we're recovering from these bioreactors over time, we find that early on, we see growth of quicker growing organisms. So in this case, this would be an example of Enterococcus faecalis. But at later time points during cultivation, we actually see a return of dominance of the original species. And this is also true for community state types 2. CST4 is much more unstable, which is actually pretty reflective of the human vaginal community state type 4 as well. So we are able to cultivate many of the organisms from there, but they tend to drift more as we progress in the experiment. And what we've done with these communities in this model system so far is really ask, how does composition of the vaginal microbiota dictate pathogen invasion? And so in these experiments, we take matched bacterial or vaginal communities matched for their bacterial composition. We allow these communities to stabilize over 5 days and then challenge them with a pathogen and then see what happens. And so in these model systems, we see that some of the communities permit pathogen invasions, whereas others do not. And you can see here, matched on composition, one community permitted invasion in both cases, really suggesting that it's not just composition alone. There's something more complex. And so we're really beginning to try to tease that apart in this model system. Another model system that incorporates the host is these human vaginal organoids, which is something that we've started over the last couple of years, and really something that we're excited about because we can now take these human vaginal communities that we're growing and apply them to human vaginal epithelium. And these organoids are developed from healthy human donor scrapings. We propagate the stem cells into a high density in matrigel, and then we plate them onto transwells and differentiate them at the air-liquid interface to allow for access of bacteria to the surface as well as secreted compounds. And this is a huge, massive effort by multiple grad students, including Sammy and Claire, as well as Gwen and Holly, and a collaboration with Dr. Sarah Blute at Baylor Organoid Core. And so what we've seen is that these organoids express high levels of proteins common in the epithelium, such as keratin 19 and MUC1. We've also seen that they differentiate into the stratified squamous epithelium, which reflects the vaginal epithelium seen in humans. And they also are really tight barriers. So this is a really high electrical resistance tier, suggesting again that we're forming an actual epithelial barrier. And we've also looked at transcriptional profiling of these organoid lines using bulk RNA-seq. And when we analyze this with human database transcriptional profiling, we see that these vaginal organoid lines cluster with human vaginal reference tissue separately from other human reference tissues, including other tissues of the reproductive tract, really suggesting that we're modeling the vaginal epithelium. We've also shown that they tolerate bacteria quite nicely, as shown by LDH release. And we've also looked at cytokine production of these organoids. And interestingly, we actually see that organoids exposed to intact human vaginal communities, these communities show suppression of inflammation without reducing cytotoxicity, suggesting that the role of a healthy vaginal microbiota may be through mediating immune suppression. We've also looked at how these organoids respond to various species challenge. And so here I'm showing you gene expression data in response to extrapathogenic E. coli. And we see multiple genes upregulated and downregulated, no surprise. Some of these genes are involved in immune signaling. And so we see interferon, IL-6, and TNF all upregulated in response to E. coli. This would be something that we'd expect for a pathogen. Conversely, when we look at a commensal like Lactobacillus crispatus, we see some similar pathways in terms of interferon responses, but we actually see decreased expression of genes associated with IL-6 or TNF alpha, again suggesting this bimodal spread of increased inflammation in response to pathobionts and a suppression of inflammation with commensals such as Lactobacillus. But this is really just a starting point. These are model systems that we're really excited about, and we're just beginning to tease them apart mechanistically. And so we have many more steps ahead of us. And in closing, I'd just like to thank members of my lab that have contributed to the development of these models, my collaborators and funding sources, and I really look forward to the discussion. Thanks so much.